class we had talked about, or we had discussed about growth phase. That means growth phase is divided into two parts: pre-vitellogenesis growth phase and vitellogenesis growth phase, right? That means we have studied about the yolk synthesis, vitellogenesis, and the activities which occur before the vitellogenesis. But we say during the growth phase only differentiation of the oocyte also occurs. That means primary oocyte was formed during the growth phase, and in this primary oocyte yolk is formed, growth is occurring, but simultaneously with the growth differentiation is also occurring. So at the stage of primary oocyte only, in the process of oogenesis, differentiation occurs. And in the spermatogenesis we have observed that differentiation had occurred after the formation of spermatids, right? So in that, differentiation was a post process, right? It occurred after the spermatid formation, but in this, during the growth phase only, differentiation occurs. Now we will discuss how the oocyte is differentiated. So firstly, establishment of axis, right? As we have already discussed that when we see the oocyte structure, this is the main axis, or we can say vegetal animal axis, right? And this is the animal pole, this is vegetal pole. So when we talk about the establishment of axis, there are three theories regarding the establishment of axis, that how the axis in the egg is, or we can say in the primary oocyte is established, right? First theory is entry of nutrition. Second theory is entry of oxygen, and third theory is every cell has character of polarity, right? Yeah, every cell must have character of polarity. So these are three theories regarding the establishment of axis. First theory says that entry of nutrition. That means scientists defined that the axis, or the point from where the nutrition or the food material enters the egg, is the vegetal pole, right? That means the part from where the nutrition enters the egg is the vegetal pole. As we have already discussed that nutrition or yolk particles are formed outside the egg and they are transported to this area, right? To the egg area. So we are seeing from where the nutritive substances are entering, that axis or that pole is called as vegetal pole. Why they said this? Because yolk is mostly present at where? Vegetal pole. So yolk is a kind of nutrition which enters inside this. So on that basis they discussed and they said that this is the vegetal pole on the basis of the entry of nutrition or nutritive substances, animals, right? But this theory was disregarded or it was not accepted. Why? Because when we talk about mammals, we say that from the whole side of the egg, nutritive substances can come into, right? So every pole cannot be called as vegetal pole because entry can occur from everywhere in mammals. So this was not accepted. Second theory was related to entry of oxygen and it was given by Child, right? Child means it is the name of scientist only, right? So Child gave this theory and he explained that the area from where the oxygen enters is the animal pole, right? That means from where the oxygen is entering inside is defined as animal pole. And he also concluded that nearby this animal pole, blood vessels or blood transportation must take place because from there only oxygen can enter through this. So on this basis it was called as animal pole. Then third was given by Balinsky, right? And he said that polarity is character of every cell, or we can say every cell must have polarity, right? Okay? We can define it in either way. So he said that polarity is the character of every cell, and on the basis of yolk deposition, whether yolk is deposited at the vegetal pole, so this pole is referred to as vegetal pole where the yolk will be deposited or will be present. And animal pole is the free pole where yolk deposition is not there. At the place of animal pole there will be nucleus and cytoplasm present and active division occurs there. And an imaginary line which divides them is called as main axis, or we can say axis, or we can say animal vegetal axis. So he just concluded this, that where yolk is present is the vegetal pole and where yolk is not present is the animal pole. Right? Now, second was establishment of symmetry. Right? First was axis, second was establishment of symmetry. So, during the symmetry establishment, some scientists concluded that where the RNA is present in large amount, that side will form the dorsal side, right? And the side opposite to this side will form the ventral side, where RNA content will be very low, right? And secondly, they also concluded that bilateral symmetry is formed in mammals and amphibians, right, on the basis of yolk content. Thirdly, he also said one thing as well, that in frogs, gray crescent area is present, and this gray crescent area defines the symmetry of frog. Right? And in some organisms, some scientists concluded that the path from where the sperm enters the egg, that region also defines the symmetry or establishes the symmetry of the egg. Right? So in this way, establishment of symmetry was described, that where RNA content is more, that side will be the dorsal side and opposite side will be the ventral side of the embryo. Now, firstly we discussed establishment of axis, entry of nutrition, from where the nutrition is entering will be called as vegetal pole, entry of oxygen, from where the oxygen is entering, that pole will be called as animal pole. Thirdly, polarity is the character of every cell. That means he also described that if we take the nerve cell, then nerve cell has dendrite region, that is just nearby region, and has axon region, which is farther away region, right? So it has different polarities. So in this way he said that every cell is polarized. So as a result, egg cell will also be get polarized on the basis of two poles, right? Now establishment of symmetry, RNA content is high, then it will be dorsal side, where bilateral symmetry is present in mammals and amphibians, where low amount of yolk is present, and the gray crescent area of the frog defines the symmetry. Now we will come to third point of the differentiation, and this third point is we can say cortical differentiation. The cortical region of the egg is also differentiated, and it is differentiated into two parts. The outermost region is called as external region, right? And the innermost region is called as inner region, or we can say endoplasm. So, external region is somewhat solid, right? And we can say this thick region, 2 to 3 mu diameter it has. So it is a thick region, solid region, right? It contains microtubules in it, right? Microtubules are present in this, as well as actin-like proteins are also present. Actin-like proteins are also present in this region, and these proteins are of tensible nature, or they are called as tensible proteins, TP, right? They are highly tensile, so they are called as tensible proteins. So what we can say is that this region is solid, thick, 2 to 3 mu in diameter, microtubules are arranged, actin-like proteins are present, and they are of tensible nature, tensile proteins, and this region, this external region provides shape, right? Provides shape to the egg. Now, when we talk about the inner region, this inner region is called as endoplasm, right? We define it as endoplasm. It is just present below the ectoderm or external region, right? And it is somewhat, we can say, thin region, right? And it is not that much solid. It is a kind of semi-solid structure. It comprises organelles, right? Organelles are present here, like mitochondria, Golgi, bodies, endoplasmic reticulum plus nucleus. All these organelles are present here. And apart from organelles, here cortical granules are also present. And these granules are arranged in this endoplasm region, right? These are the granules like structure and present in cortex region, that's why called as cortical granules. Later on they come to the peripheral part, but they are formed here only, right? They are formed in the endoplasm region and later on come to the peripheral part. They are formed from Golgi bodies, right? Golgi bodies help in the formation of cortical granules, and in the cortical granules mucopolysaccharides are present, right? They comprise mucopolysaccharides in them. That means they have mucopolysaccharides in them. And second thing, cortical granules help in the fertilization process, right? They help in the fertilization process and they are present in mammals like rabbit and humans. They are present in some amphibians, not all, right? They are absent in mouse, guinea pig. They does not contain cortical granules. So cortical granules are present in only some mammals like rabbits and humans, or some amphibians also contain these type of structures, right? And they help in the process of fertilization. They help in the formation of fertilization membrane, we can say. Fertilization membrane is formed due to these structures only. We will discuss this later on in the fertilization chapter, that how fertilization membrane is formed. But for now, it is just like this, that cortical differentiation occurs in two parts, external and inner. External side is solid, contains microtubules and actin-like proteins, and it is thick. Just below this lies endoplasm, inner region. Here semi-solid or thin structure is present, that is not much thick or solid, right? Organelles are present. Apart from organelles, cortical granules are formed here, and later on they are shifted to the peripheral part. These are formed by the Golgi bodies and they contain mucopolysaccharides, right? And then fertilization membrane is formed by these granules, and they are present in rabbits, humans, and in some amphibians as well. So this was differentiation in oocyte, establishment of symmetry, axis, and thirdly cortical differentiation. So here we complete the growth phase. Growth phase is the largest phase which contains two types of things. First the vitellogenesis part, pre-vitellogenesis part, and then differentiation. So in growth phase, with the formation of yolk, differentiation also occurs. Now we will discuss the types of eggs. Right? There are different types of eggs present and they can be divided on different bases. So first will be on the basis of amount of yolk, right? On the basis of amount of yolk, egg can be egg can be of 4 types, right? When we say the egg is of alecithal type, that means there is no amount of yolk, or amount of yolk is just nil, right? Or yolk is just absent there. The name itself suggests lacithal means yolk and A means absence of yolk, right? So in these type of eggs, yolk is completely absent, and the example of these types are mammals, most probably mammals, eutherian mammals, which are true mammals, right? Which are not oviparous, viviparous type of organisms. That means they produce direct young ones and they do not produce eggs outside, right? So, in mammals, in eutherian mammals, alecithal type of eggs are present where amount of yolk is completely absent. Second type of eggs are microlecithal or oligolecithal. In these eggs, micro means very small amount, right? Or here, very small amount of yolk is present. Right? So these type of eggs contain very small amount of yolk, or we can say in the form of granules they are present. So when we come to the examples of these type of eggs, example is lower invertebrates like branchiostoma, tunicates like herdmania and echinoderms. Right? So, they are present in lower invertebrates, branchiostoma and tunicates, which are lower chordates, and echinoderms, right? When we talk about mesolecithal type of eggs, they contain medium amount of yolk, right? Or we can say intermediate amount of yolk is present in these type of eggs. So they are called as mesolecithal. And in these type, example is amphibians, most probably frogs come under this category. Now lastly, megalecithal or polylecithal type of eggs, they contain very good amount of yolk, or we can say large amount of yolk is present in these type of eggs. And examples are reptiles and birds. Alright, reptiles and birds contain megalecithal, or we can say large amount of yolk in their eggs. So, on the basis of amount of yolk, 4 types of eggs are present. Now comes the second category. On the basis of distribution of yolk, right? Like how the yolk is distributed, on this basis also eggs are divided. So they are again of different types. Right? So they are again of 4 types. When we say the eggs are of isolecithal type, iso means the distribution of yolk is over the entire egg. So when we draw the diagram, we see or we observe the distribution completely over the whole egg. So this type of egg is called as homolecythal or isolecythal. That means we can say distribution of yolk is completely over the whole egg. So that's why we call it as isolecythal or homolecythal type. And these eggs are of microlecythal nature, right? That means very small, very little amount of yolk is present and it is distributed in the form of granules. So this is yolk, right? And this is isolecythal egg. Always when we draw diagram, we should label it, right? This is egg. So, examples of isolecythal can be same examples of microlecythal, like we can say echinoderms, right? Branchiostoma, tunicates. Right? Tunicates are those organisms which contain tunic membrane around them or test membrane around them. And tunicates example is Herdmania. Best example is Herdmania. You will study later on. Then second category is moderately telolecythal type of eggs. Here we can say that yolk is present at one pole, right? And cytoplasm is present at the other pole. So they are called as moderately telolecythal because of the one reason that firstly we say yolk is distributed over the one pole of the egg, and here the cytoplasm is not as much separated, right? So they are called as moderately telolecythal. Secondly, when we talk about telolecythal type of eggs, so here we say again yolk is distributed over one pole of the egg. Again the same thing comes here, but here what happens is yolk is separated from the cytoplasm. Easily it is separated from cytoplasm, and here one structure like cytoplasmic cap is present, right? Cytoplasmic cap-like structure is also present. So when we talk about the telolecythal type of eggs, they are of polylecythal or mesolecythal category, right? So in this example is frog or amphibians comes under this, right? Now, when we talk about the centrolecythal type of eggs, here the yolk is present at the center, completely at the center, so they are called as centrolecythal, right? is present at the center of the egg. They are centrolesical, right? And these type of eggs are again of which type? Polylesical type. That means they contain large amount of yolk in the form of platelets, right? So here yolk platelets are present. And when we talk about the example is insects in this category, right? So on the basis of distribution of yolk, again they are divided into 4 types, right? Now next, third category of eggs, and third category is on the basis of shell, right? On the basis of shell, again they are divided into 2 categories. First is so, if shell is present outside the egg, shell is what? A structure of hard covering like structure which covers the egg from outside, and it is made up of calcium carbonate, right? And it has some small pores which helps in the respiration, that means exchange of gases CO2 and O2. So it is made up of calcium carbonate like structures. So if they are cleanoic eggs, that means shell is present in them, and these type of eggs are laid on the ground, right? On the terrestrial surface they are laid. So they need shell to prevent or to protect the embryo, right? Because they are laid on hard surface, so embryo can get damaged by laying, so shell is a must thing. So cleanoid eggs are the eggs in which shells are present, and examples are reptiles and birds, right? So non-cleanoid eggs are those eggs in which shell is absent. So they can be laid on the water where protection is not as much needed, right? So example of these can be mammals, frogs, amphibians, etc. Right? So on the basis of shell, again they are categorized into 2 categories. Now fourth type of eggs are divided on the basis of embryonic development, right? On this basis as well, eggs are divided into 2 types. Determinative or mosaic type of eggs are those eggs in which future is already determined, right? Or future is determined. Future can also be called as fate, that means in which fate is determined, right? So if here embryo is present and all the cells have certain future. So cells outside from here. So certain structures will not be formed or certain structures will be missing after removal, right? So if we remove one cell from the developing embryo, because the fate is already determined, so the structure which that cell will be forming will not be formed or will be removed, will be absent there in the organism after the growth, right? So this type of eggs are called as determinative or mosaic type of eggs, in which future is already determined of each and every cell. An example of these are lower invertebrates, lower chordates, etc. Right? Second category is indeterminative or regulative. So here future is not determined of each and every cell, right? And so if we say we remove one cell from this embryo, right? If we remove one cell from this embryo, then also all the parts will be formed. All the parts will be formed even after the removal of the cell. Or blastomere, right? So we are very much clear that their future is not determined. So even after the removal of the cell as well, all the body parts will be formed. So it is called as indeterminate. That means the adjacent cells will give signals regarding that structure and all the parts will be formed. That means cells are there communicating with each other and they target one cell to form that structure. Right? So they are called as regulative. They are regulating in themselves to form the whole organism. So on the basis of embryonic development as well, again we divide the cells or we divide the eggs into two types, determinative and indeterminative type. Now we will see the significance of oogenesis. Right? What is the significance of oogenesis? Or we can say importance of oogenesis. here meiosis first occurs, which reduces the maternal characters or maternal chromosomes to half. This is an important event with respect to oogenesis, right? So maternal characters are reduced to half, or we can say haploid eggs are formed. Restoration of chromosomes occurs during the zygote formation because both the gametes are haploid. So when they combine during the fertilization or fuse to an or diploid organism is again restored. So chromosome numbers are reduced during the meiosis, but during the zygote formation they are again restored, right? Yolk synthesis occurs during the oogenesis process, and it helps in the nourishment, nutrition, and provides certain substances for the formation of proteins and structures, right? Now, egg membranes are also present and formed during oogenesis process. They help in the prevention of polyspermy or to fertilization. Apart from these, they also help in certain other activities like floating of egg on the surface like these, right? And even in the protection as well from the environmental stimuli or factors. Now, cortical granules are also formed during the process of oogenesis, and these granules help in the formation of fertilization membrane during the fertilization process. So it is also of significance and great significance we can see, because this membrane must be formed during fertilization, right? Lastly, we talked that unequal division occurs during the meiosis first and meiosis second. It is really of significance because it helps in restoration of yolk or food material, reserved food materials. That's why unequal division occurs, and here from we can say one primary oocyte, one secondary oocyte is formed, and from one polar body is formed, and polar bodies get degenerated, we can say. So we can say from one primary oocyte, 4 cells are formed or 4 structures are formed, but only one is functional, which is the mature ovum or ooted. Rest 3 degenerate and are not used in the process of oogenesis. So these are the significances and importances of oogenesis.